Lipids are the most efficient form of energy storage in animals. Other biomolecules can be classified according to what are the functional groups present. For example, carbohydrates are classified as polyhydroxyketones or polyhydroxyaldehydes. This is not true about lipids. The family of lipids will contain ester bonds, amide bonds, polyesters, ketones, alcohol groups, and many others. Lipids come in a variety of molecular structure and with diverse functions. For example, triacylglycerols are energy storage. Steroid hormones act as chemical messengers. Prostaglandins and eicosanoids are local hormones or local mediators that act in the same tissue where they are synthesized. Other lipids, such as phospholipids from glycerol and phospholipids from sphingosine, and cholesterol are important parts for membranes. The only thing that all lipids will have in common is a high content of carbons and that they do not mix with water. Lipids can be classified in two different groups. Those that by hydrolysis you can produce a free fatty acid and those that cannot be hydrolyzed. For example, a wax is a lipid that contains one ester bond with very long hydrocarbon chains, and it can be hydrolyzed to produce a free alcohol and a free carboxylic acid. In a sphingolipid, we can hydrolyze amides bonds to produce a free amine and a carboxylic acid. Among the non-hydrolyzable lipids, we find steroids, prostaglandins, and some vitamins. These cannot be hydrolyzed because they do not contain an ester, an amide, a phosphoester, and they cannot release a free fatty acid. For example, animal fat contains 3 ester bonds. Strong solution of potassium hydroxide or sodium hydroxide will cleave these ester bonds to produce 3 free fatty acids and 1 unit of glycerol. Because cholesterol does not contain any esters or amides, it cannot be hydrolyzed. The simplest lipid is a fatty acid, which can be defined as a long hydrocarbon chain carboxylic acid. Among its properties, we can describe that fatty acids are, most of them are unbranched, they can be saturated, unsaturated, polyunsaturated, and those that we call omega-3. Because they are synthesized from acetyl coenzyme A, they are synthesized in a fashion of 2 by 2, and most of the fatty acids contain an even number of carbons. The number of carbons ranges from carbon 10 to C24, 26. When we discuss metabolism, we are going to pay close attention to the molecule of acetyl coenzyme A, which is the one used to put together 2 carbons at a time to make fatty acids. Stearic acid is a fatty acid because it has a long hydrocarbon chain. It is 18 carbons long, and it is saturated because it does not have any carbon-carbon double bond. It is unbranched because all of the carbons are bonded only to hydrogens and they do not have any methyls or ethyl branches. Due to the high content of carbons, these molecules are water insoluble. It is the only thing that all lipids will have in common, and that is that they do not mix with water. Branched fatty acids are rare in plants, but they are found in the membranes of some bacteria and archaea. Besides saturated fatty acids, all single bonds, we also find unsaturated fatty acids and polyunsaturated fatty acids. The difference between those two is relevant to membrane structure. The higher the number of double bonds on a fatty acid, the lower the melting point. We can see the stearic acid that has 18 carbons long has a melting point of 69. When we have linoleic acid with 2 double bonds, same number of carbons but lower melting point. This will be part of oils, and stearic acid can be a solid. Why is this? The reason is that saturated fatty acids can have higher number of London dispersion forces or van der Waals interaction, hydrophobic interaction between the hydrocarbon chains. In contrast, when there is unsaturation, the double bond produces kinks, bends in the hydrocarbon chain, giving less interaction between those hydrocarbons. Membranes with a high content of polyunsaturated fatty acids in their phospholipids will have less interaction when they are facing one another, forming a lipid bilayer. Among the polyunsaturated fatty acids, we also find those called omega-3. And that is, if we count using IUPAC, the carboxylic acid carbon, that will be carbon number 1. But according to the omega-3, the carbon number 1 is the last. So counting from the omega, which is the last carbon, we will find a carbon-carbon double bond on carbon number 3. Omega-3 fatty acids are essentials because they cannot be synthesized in our bodies, and they are found in fish oils and plant seeds. In our discussion of organic chemistry of carboxylic acids, we have seen that the reaction between a carboxylic acid and an alcohol in the presence of a catalyst at high temperature produces an ester upon a removal of one molecule of water. If in this reaction we use a fatty acid, or let's say a very long hydrocarbon chain carboxylic acid with a very long hydrocarbon chain alcohol, which must be always a primary alcohol, the product is called an ester. These esters are called waxes. They are esters of high molar masses fatty acids and high molar masses alcohols. Waxes represent one of the most hydrophobic classes of lipids. The extreme water insolubility allows waxes to serve a protective function. Aquatic birds have a water-repellent waxy coating on their feathers. The surface of fruits and some leaves are also covered with some waxes. Waxes tend to be the hardest from all of the lipids because they have more hydrocarbons than any other, having a long hydrocarbon chain carboxylic acid and a long hydrocarbon chain alcohol. Chemically, fats and oils are esters of glycerol and fatty acids. Animal fats and vegetable oils are the lipids most frequently found in nature. They are synthesized by dehydration reaction, forming 3 ester bonds in the molecule of glycerol. The product contains 3 ester bonds that can be hydrolyzed during metabolism by enzymes named lipases. The difference between a fat and an oil is the content of carbon-carbon double bonds. In a fat, the R groups present in the triglyceride are mostly saturated fatty acids. In oils, the structure of the triglyceride contains R groups that are mostly polyunsaturated fatty acids. The bends around the carbon-carbon cis double bonds decreases the interactions between the hydrophobic hydrocarbon chains. Another important family of lipids that is not hydrolyzable is that of eicosanoids. The word eicos means 20, which means that these are lipids derived of polyunsaturated fatty acids 20 carbons long. Prostaglandin, leukotriene, and thromboxane are all derivatives of arachidonic acid. They play roles in the inflammatory response in joints during arthritis, the production of pain and fever in disease and injury, the regulation of blood pressure, the induction of labor, the regulation of wake and sleep cycle, and in allergic and asthmatic reactions. Steroids are non-hydrolyzable lipids that contain the steroid ring structure, that is, 4 fused rings A, B, C, and D. Cholesterol is the major steroid found in animals. Plants contain very little cholesterol but do not contain related compounds in their membranes. Among the derivatives of cholesterol or steroids, we find the hormones cortisol, aldosterone, testosterone, and progesterone. Because cholesterol has a hydrophilic hydroxyl group and a hydrophobic part, it is found in animal membranes. These complex lipids that contain phosphate groups will be discussed in a separate video in the discussion of membranes.